you. Thank you, Paul. So I have the honor and the pleasure and the happiness to introduce not only a good friend of mine, but an incredible scientist, Professor Ranulfo Romo, from the University Autónoma of Mexico and el Colegio Nacional. And Ranulfo is very well known, so Paul asked me to do a very short introduction and to keep all the things so brief as possible, and that is very easy with Ranulfo because it's worldwide known, particular because he has done incredible experiments and advances in the field of decision making with monkey neurophysiology. He was originally trained as a medical doctor and then as a neurophysiologist with probably one or with the father of the neurophysiology in the States, Moncaster. And then many years after that and many postdocs after that, he came back to Mexico and started his own lab. And I hope that he tell us some anecdotes about that time because it's very funny. But the most important thing is really the type of experiment that he was doing during the last 20 years. I think it's one of the unique labs able to do multiple recordings in multiple seats, which is absolutely a paradise for theoreticians at least, like you and me. So we are very happy to have you here and happy to hear your lecture.
Thank you. It's a pleasure to be here, and of course, to see old friends like Gustavo and meet and have new friends like you is a great opportunity to me. I made a very long trip from Mexico City to come and be with all of you, and please feel free to talk to me, interrupt me during my talk if something is not clear enough. Second point, I was not planning to say some words about Werner Mountcastle. It takes almost a generation to forget everything. But Werner Mountcastle was not only my mentor, was mentor of many neuroscientists, not only in the United States, but also across the world, and laid down many basic principles for studying brain organization, how the brain processes sensory inputs, and how these inputs are used by brain cells to produce cognitive functions, something like that. He died early this year at the age of 96, and he was a very incredible man, and as a mentor, as a scientist, and as a person by his own. So in some way, I'm only an extension of Werner Mountcastle, and I want to believe that you will be also an extension of many of your mentors. So, and you will have to carry on your shoulder traditions and projects, train new generations, and to continue our project is, for example, just not only to understand the brain by itself, but also to all the products of the brain, like technology and life in a general term. So, for over the years, I've been very interested in understanding decision-making. The word decision-making is a very nice one, it's very popular at this moment, but in my old times and the times of Werner Mountcastle, it was called simply like sensory to motor transformation. So the word sensory to motor transformation is simple transformed now to a very fancy word, which is decision making, which is, as I said, very popular. And in fact, this became very popular by the end of the 80s, when I was just returning to Mexico, and I was planning to do a series of experiments using the somatosensory system as a model. I am not a somatosensory man. I used the model as a model. By that time, Bill Newsom, a very well-known man, scientist from Stanford University, started to use the visual system to understand how, at least the visual system, processes sensory inputs and transform that inputs into decision making. And so he made a very terrific contribution to science, which was taken over by Mike Shadlin over the last 15 years or so. So it was more or less the same idea, Bill Newsom in the visual system, myself in the somatosensory system. And we carried over a number of experiments in such a way that it was clear to us that the central question was how a sensory input in the brain is represented by brain cells. It's like a transformation, it's a neural copy of the stimulus into the cerebral cortex. And the question was, where and how? So it was quite easy to us that it was made by these sensory areas. If you were using the somatosensory system, very simple stimulus, as you will see in a few minutes, you see how brain cells encode and respond to the stimulus. And using very simple statistical tools, you can decode something from the responses of brain cells and relate those responses to the stimulus parameters. But decision making is a little bit more complex because our brains have experience keeping memories, long-term memories or short-term memories. And so there is a combination between those early representations with something stored in long-term or short-term memory. So somehow, these two have to the internal representation of something, goals, motivation, whatever you think about, have to be combined, and that product is what is used for instructed the motor apparatus and give a response. And in total, all this is called decision making. Most of the labs, including myself, use only one sensory modality in animals in particular. It's more easy to do that in humans because you don't have to train them, or sometimes you have to train a little bit. When we were doing these experiments using the somatosensory system, it was clear to us that it was a very large network that was engaged in decision making. And the question to us at the time was whether this was a general principle for all modalities, or whether the network, the brain network that we were studying using one sensory modality was devoted, was very specifically engaged in somatosensory decision or visual decision. So it was clear to us that we needed to use another modality, a couple of modalities at least, and train the animals to treat the sensory inputs coming from those sensory modalities to produce decision making. So I'm going to speak about this. It's been a very difficult experiment because, as I said, monkeys do not know what we want from them. And it's been clear to me over the last years that when monkeys go to the lab to be trained to do something, it's exactly the same what the students do when they go to the university. At birth, you know nothing about engineering, medicine, law, or something like that. So you go to the faculties of engineering to train your brain to become engineer or a medical doctor. So your neural circuits are trained in such a way that when you are an engineer and you are facing a problem, that input trigger the network. And if you trigger the network, there is something has to be knowledge about something has to be stored in a network trained previously. It's exactly the same what happens with monkeys when they go to the lab. They are trained for something that they didn't know. But at the end, when they are confronting the task, the monkeys are able to do that in such a way that when a new student comes to my lab, I always take a naive monkey, and I train both. So the monkey learns the task, the student is trained to train the monkey and learn the task too, but over the few weeks or a month or two, the student becomes a little bit mad against me because the student believes that I am treating him or her like a monkey. It's not. They both came to the lab to be trained. So I'm going to speak about this. Of course, at the moment, we don't have a clear idea how something is this knowledge stored in the network. And it's an important problem, not only right now, but in the future, to understand how our brain keeps experience and uses it in an intelligent manner, not always, to make decisions about something. So I'm going to speak about this tiny problem, which is where and how in the cerebral cortex the single neurons process more than one sensory modality during perceptual judgment. The experiments had been done, but Pepe Vergara, is a PhD student in my laboratory, is extremely intelligent, as intelligent as the monkey. And his main task is to train animals to do what I'm going to present and to decode, use simple statistical tools to decode information from the responses of brain cells as the monkey carries the task. So in the first part of my talk, I will introduce the model, the behavioral model we use in our laboratory. And in the second part of my talk, I will document this. And please feel free to interrupt me if something is not clear enough to understanding something about what I'm talking about. So this is a cartoon of a rhesus monkey, which sits in the laboratory in a special chair designed for it. And as I said, we use the somatosensory system as a model. We deliver very tiny parametric signal, very well controlled, delivered to the skin in such a way that we stimulate cutaneous mechanoreceptors and which are transduced by receptors, and these transduced signals are transmitted by the nervous to the upper part of the spinal cord, then to the thalamus, and finally to the cerebral cortex. And in the cerebral cortex, there is a tiny representation of the stimulus parameter, instantaneous, it lasts very few, half a second or something like that. And magically, these representations is transformed into voluntary actions. And believe me or not, but if somebody discovers how this tiny representation, this touch representation, is transformed into a voluntary action, it certainly wins a Nobel Prize. So, in spite of this lack of knowledge, I want to continue speaking about this, and I have to introduce how we do this experiment. So, goes down, is something like the tip of a pencil, touches the skin, makes a very tiny indentation, about 300 microns, protrudes the skin, produces a mechanical stimulus, is transduced, and sends information to the cerebral cortex, which arrives in about 25 milliseconds, very rapid. Then, in response to this, the monkey reacts, placing his hand. It's a voluntary action, because if the monkey doesn't want to do it, it doesn't do, simple like that. Places its hand, the computer detects that the stimulus is there, and the hand is touching the skin. And then, suddenly, it comes superimposed a tiny vibration, which is in the flutter range, which can be from 5 to 40 hertz. And from trial to trial, the stimulator picks one frequency and delivers into the skin. All these stimuli are suprathreshold, about 7 times the threshold, with the threshold is about 7 microns. It's something for frequency to frequency. We have to make it that all stimulus frequencies are treated equal, of the same magnitude, stimulus magnitude, in such a way that monkey is concentrated in the number of pulses presented during half a second. So the monkey senses that, but it can come another stimulus. The monkey never knows whether there will be a tactile, vibrotactile, delivered here, or something coming to the ear by acoustic stimulus, which is also a repetition simulating more or less the same trains delivered to the skin. So the monkey never knows whether tactile or whether is acoustic. It can be any frequency, as I said before, for either tactile or acoustic. So the monkey has to use all its cognitive resources, attentional resources, to be attentive to any of these two stimulus. Then the stimulator stops, and there is a gap of about 3 seconds, in such a way that the monkey has to store one of the two stimulus in working memory. Working memory is a special cognitive capacity that animals and you and I have, I think. Otherwise, we couldn't understand something when we are speaking or when we are listening to someone like me. So you have to store in short-term memory something in order to keep going and understanding where you are and what's going around you, or feeling something. Then, it can be, again, a tactile versus tactile, or acoustic versus acoustic, or cross-modal, and compare the second one against the first. And if you do a comparison between the second against the first, unavoidable, you have to make it the fresh stimulus, this one, against the trace kept in working memory. So that it's a very rich task. It's very simple, very parametric, but you can trace not only the stimulus representation in the early sensory cortices, but also where in the brain cells store parametric information in working memory, and how the two are combined in order to make a comparison and, of course, decision-making, and respond whether, for example, the second is higher for one modality or whether the second one is lower than the first. And with that, you can apply this simple protocol. For example, in a trial, tactile versus tactile, and when the differences between the two are very large, for example, 8 hertz between tactile versus tactile, first and second, you have a large percentage of successful responses. And surely the monkey is doing this. Or, for example, you can do this. The first is tactile and the second is acoustic. And again, you can see that the monkey is quite good for that. Or for example, the first can be acoustic and the second acoustic, and you can see the numbers indicate the percentage of successful responses. Or for example, acoustics versus tactile, and see how percentages are very large, indicate that the monkey is doing it in a very easy way. If you apply this protocol, you can go and try to understand all the processes associated to not only for the same modalities or cross-modals, and try to understand all the process associated. But you can do more than this, and this is made by psychophysical test in which you can get the thresholds. For example, these are simple cross-modal, the psychophysical curves, in which the monkey is comparing the acoustic versus the tactile, and here are the probabilities that the second stimulus is higher than the first. The colors indicate when, for example, the second one, which is acoustic, is higher than the first. This is the psychophysical curve, and this is for the opposite, in which the second is the first is you are manipulating the first stimulus, and then the second one is fixed. And it's exactly the same. The first can be acoustic, the second is tactile, and you will see that the shape of the curves are very similar. These are the psychophysical thresholds, which is no more than the 75% of the upper limb of the curve and the 25% of the lower limb. Divided by 2, you can get the thresholds, and you will see that they are very similar. So all these trials, easy and very difficult trials, are mixed together, delivered in a single run, which is a very hard experiment because the monkeys have to be attentive during this psychophysical test for almost more than an hour, concentrated, because it's something like 600 trials in order to get all these curves. When you have this, then you can pose the questions, the neurophysiological questions. So you have a very nice model in which you are clearly sure that the monkey is doing what you want to do, or what the faculty of medicine or engineering wants from you when you study one of these. And the first question is very simple and came to us not as something very new. It's simply that there came a wave of papers by the end of the 80s, beginning of the 90s, beginning between 2000 and up to now, in which many people started to say that early sensory cortices had the capacity to process more than one sensory modality. Not only its principal modality, but also another. For example, the visual cortex had not only the capacity to process visual inputs, but also tactile, acoustic. Or the primary auditory cortex had not only the capacity to process acoustic inputs, but also tactile and visual, and for the somatosensory cortex as well, that it not only processes somatosensory input, but also acoustic and visual. And to us was odd, because it's a very old dogma. If you go to textbooks of neurophysiology textbooks, they will tell you that information from the senses are processed, but sensory cortex are unimodal, only processing one modality, visual for visual, auditory for auditory, acoustic input, somatosensory, parasensory input. But it was even more odd to us that the people started to say that not only had the capacity to process more than one modality, not only its principal modality, but also had the capacity to keep in working memory more than one sensory modality. also challenging an old dogma that downstream areas to sensory cortices were the responsible for keeping sensory inputs in working memory. All these stimuli are suprathreshold, about 7 times the threshold, with the threshold is about 7 microns. It's something for frequency to frequency. We have to make it that all stimulus frequencies are treated equal, of the same magnitude, stimulus magnitude, in such a way that monkey is concentrated in the number of pulses presented during half a second. So the monkey senses that, but it can come another stimulus. The monkey never knows whether there will be a tactile, vibrotactile, delivered here, or something coming to the ear by acoustic stimulus, which is also a repetition simulating more or less the same trains delivered to the skin. So the monkey never knows whether tactile or whether is acoustic. It can be any frequency, as I said before, for either tactile or acoustic. So the monkey has to use all its cognitive resources, attentional resources, to be attentive to any of these two stimulus. Then the stimulator stops, and there is a gap of about 3 seconds, in such a way that the monkey has to store one of the two stimulus in working memory. Working memory is a special cognitive capacity that animals and you and I have, I think. Otherwise, we couldn't understand something when we are speaking or when we are listening to someone like me. So you have to store in short-term memory something in order to keep going and understanding where you are and what's going around you, or feeling something. Then it can be, again, a tactile versus tactile, or acoustic versus acoustic, or cross-modal, and compare the second one against the first. And if you do a comparison between the second against the first, unavoidable, you have to make it the fresh stimulus, this one, against the trace kept in working memory. So that it's a very rich task. It's very simple, very parametric, but you can trace not only the stimulus representation in the early sensory cortices, but also where in the brain cells store parametric information in working memory, and how the two are combined in order to make a comparison and, of course, decision-making, and respond whether, for example, the second is higher for one modality or whether the second one is lower than the first. And with that, you can apply this simple protocol. For example, in a trial, tactile versus tactile, and when the differences between the two are very large, for example, 8 hertz between tactile versus tactile, first and second, you have a large percentage of successful responses. And surely the monkey is doing this. Or, for example, you can do this. The first is tactile and the second is acoustic. And again, you can see that the monkey is quite good for that. Or, for example, the first can be acoustic and the second acoustic, and you can see the numbers indicate the percentage of successful responses. Or, for example, acoustics versus tactile, and see how percentages are very large, indicate that the monkey is doing it in a very easy way. If you apply this protocol, you can go and try to understand all the processes associated to not only for the same modalities or cross-modals, and try to understand all the process associated. But you can do more than this. This is made by psychophysical test in which you can get the thresholds. For example, these are simple cross-modal, the psychophysical curves, in which the monkey is comparing the acoustic versus the tactile, and here are the probabilities that the second stimulus is higher than the first. The colors indicate when, for example, the second one, which is acoustic, is higher than the first. This is the psychophysical curve, and this is for the opposite, in which the second is the first is you are manipulating the first stimulus, and then the second one is fixed. And it's exactly the same. The first can be acoustic, the second is tactile, and you will see that the shape of the curves are very similar. These are the psychophysical thresholds, which is no more than the 75% of the upper limb of the curve and the 25% of the lower limb. Divided by 2, you can get the thresholds, and you will see that they are very similar. So all these trials, easy and very difficult trials, are mixed together, delivered in a single run, which is a very hard experiment because the monkeys have to be attentive during this psychophysical test for almost more than an hour, concentrated, because it's something like 600 trials in order to get all these curves. When you have this, then you can pose the questions, the neurophysiological questions. So you have a very nice model in which you are clearly sure that the monkey is doing what you want to do, or what the faculty of medicine or engineering wants from you when you study one of these. And the first question is very simple and came to us not as something very new. It's simply that there came a wave of papers by the end of the 80s, beginning of the 90s, beginning between 2000 and up to now, in which many people started to say that early sensory cortices had the capacity to process more than one sensory modality. Not only its principal modality, but also another. For example, the visual cortex had not only the capacity to process visual inputs, but also tactile, acoustic. Or the primary auditory cortex had not only the capacity to process acoustic inputs, but also tactile and visual, and for the somatosensory cortex as well, that it not only processes somatosensory input, but also acoustic and visual. And to us was odd, because it's a very old dogma. If you go to textbooks, of neurophysiology textbooks, they will tell you that information from the senses are processed, but sensory cortex are unimodal, only processing one modality, visual for visual, auditory for auditory, acoustic input, somatosensory for somatosensory input. But it was even more odd to us that the people started to say that not only had the capacity to process more than one modality, not only its principal modality, but also had the capacity to keep in working memory more than one sensory modality. Also challenging an old dogma that downstream areas to sensory cortices were the responsible for keeping sensory inputs in working memory. For example, prefrontal cortex had the capacity to keep visual information, for example. But now the people started to say that the primary somatosensory cortex had not only the capacity to process the sensory input, but to store it in its modality, another modality. We were shocked, us in the laboratory, and thought that we had to reevaluate this problem in our own paradigms. Of course, very often early in the morning I wake up and think, maybe they are right. The question is simple. I'm using some other paradigms, and we are not across. We are not probably dealing with different problems. Anyway, I'm going to present to you something which we've been working systematically over the last 10 years, trying to understand and give an answer to this simple question. And these are, of course, you already know the paradigm for the two tasks. This is a cartoon of the monkey brain. In blue is the primary somatosensory cortex. You go to the somatosensory cortex, you're delivering the stimulus in the third finger, in the skin of the third finger. You have to go more or less to the territory of the primary somatosensory cortex, where the inputs from the thalamus are represented, information only for the third finger. And very often, I'm asked, and how many neurons respond to the stimulus in the primary somatosensory cortex? I always say, it depends. You deliver the stimulus in the foot, and you are recording in the third finger of the hand, you will not see one single neuron responding. So somatotopy is very important. And this is about the area for the primary auditory cortex, which is inside the lateral sulcus, and you have to manage to do recordings in auditory cortex while the monkey is performing the task. Not only here, but also here. And of course, this is a very simple cartoon. We simply drive the microelectrodes into the hand area or in the auditory cortex and bring the tiny microelectrodes of about 10 microns very nearby the neuron. And you know that the neurons generate electrical signals, and we simply pick up them. We bring the electrode very close to the soma and do the recording of the cells, amplified. And not only the spikes, but also some other signals like all what's going on, local field potentials, for example, and do the analysis and make correlations between the stimulus parameters and the cognitive capacity of the monkey with the brain cells while it's performing the task. So this is all about, and you can see it's very simple. Train the monkey, bring your microelectrodes in these brain areas, and go nearby the soma and do the recordings. And that's it. And then enter in action, analysis, and trying to make sense of these responses. And this is what happens with every neuron you are studying in the primary somatosensory cortex as the monkey is performing the task. So bring the microelectrodes here, and you will see these are, on the left side, are some few frequencies for the first stimulus, which are represented here. It's tactile, delivered to the skin. This is the first. This is the second. You will see just by eye, using your visual system, that there are more pulses here than here. So you can say that this is higher than the first, just simply because you are observing the pulses. But if I deliver this and then delete it from your visual scene and then deliver the second, you have to use working memory and don't to remember that the second one is higher than the first one. And the first thing that it happens here is that the neuron respond very nice to the first stimulus in such a way that they are phase locked to the sine wave delivered to the skin. They are a little bit delayed, about 25 milliseconds, but the representation is that, no more than that. It's a stimulus parameter representation there in the responses of brain cells. The second thing that can be detected immediately is that there is nothing during the gap, during the working memory component. There are no variations as a function of the stimulus, the first stimulus frequency. And the neuron responds again to the second one, indicating more or less the stimulus parameter. And here is where the monkey has to make comparison. The monkey doesn't listen at this moment, doesn't see anything. He simply is feeling something in the fingers. and making all these cognitive computations using this representation then. But if you're recording the same neuron in the primary somatosensory cortex, you can test it whether it responds also to the other modality, acoustic versus acoustic. And you will see that there are no longer responses to the acoustic stimulus, nothing during the working memory component, and nothing during the comparison problem. At least, just by eye, this tells you that these neurons are very unimodal, responding to the tactile, not to the other modality. Okay, and this, if you use the cross-modal test, first tactile, second acoustic, you will see that it only responds to the tactile, nothing to the acoustic. And if you do the opposite test, inverse the sequence, first acoustic, second tactile, you will see that it only responds here. And this is the whole picture. I even do not need fancy analysis to show that this part of the cerebral cortex, which is the primary somatosensory cortex, is unimodal. If you want to know more about all this, there is a neuron paper published in 2010 where you can get all the details about the behavioral test, about all the literature before that paper, and all the analysis, statistical analysis used in order to prove this. Not only for the sensory representation, but also for the working memory and the decision component of this test, across all the somatosensory areas. Now, doing the same experiment, you can go to the auditory cortex and test whether the auditory cortex not only processes the acoustic stimulus with the same logic as for the primary somatosensory cortex, but also the tactile one. In fact, trials are mixed together in the primary somatosensory cortex and in the auditory cortex for the two modalities. So it's very, very nice because you can, at least in this stimulus design, task design, you can test whether one is unimodal or whether the other one is multimodal or something like that. So you will see that if you go with tactile stimulus, there is nothing during the stimulus presentation, the working memory comparison, and decision making. But if you go with the acoustic stimulus test, you will see that the neuron responds. It's very similar to the temporal order, periodic order of the repetition of the stimulus. They are phase lock, again, to the first stimulus, but there is nothing during the working memory component, comparison, and decision component. It looks like very unimodal, the auditory cortex, and forcing the network to tell in a more complex test in which the monkey has to treat the tactile and the acoustic stimulus as equal in order to make comparison. You will see that the neurons are responding very nicely to the acoustic stimulus in the auditory cortex and nothing to the tactile one. So the question is, this is the whole picture for the auditory cortex, very acoustic-oriented, responsive, and nothing with the tactile one. So, after a very big effort, we came to the conclusion, to the old conclusion, that early sensory cortexes, at least for these two cortices, of course, we didn't test the visual cortex because we had to design a trimodal test, which is extremely difficult for the animals. To do one task, unimodal task is hard. To train the monkeys to do two tests, two sensory inputs mixed together is hard. And nobody, of course, has tried with a task of this type. So if you like, it's open, where this might happen in the visual cortex, but I'm pretty sure I could bet all my money, which is at devaluated Mexican pesos right now, that I bet you that I'm totally sure, because I have done the experiment, sure, by sure. Okay, so this is my conclusion. Of course, there can be some other processes associated to this. Do you believe that the brain is only made for processing sensory input? No. More of our life is using our internal representation, something stored in working memory. The only way that Professor Gustavo Deco does his work is precisely because his brain has been trained in physics and computational neuroscience. So he can, by some mechanism unknown to, perturb that circuit train and start thinking about his work and treating information from different Ranulfo, Paul here, and I don't know where, and that, and putting together to think about and tapping his computer and using some formula and writing and thinking and thinking and thinking. And suddenly one of his students comes and interrupts him. Acoustic stimulus or visual stimulus, and its brain is treating the information, but using probably the same network in order to treat that. So this is perceptual decision making, but in his case can make decisions using his experience, is stored in long-term memory. And that is a very interesting issue, to discover all the principles that make it possible to have those. So now, but if we gave a negative, well, positive to us, a negative answer for the rest of the neuroscience community, at least for the multimodal community, were mad, many colleagues. In fact, there was a preview paper about this in Neuron, and the fellow who wrote it, he wrote it in a very strange way, it was a disgrace for the multimodal community about us. He never referred any positive result from our paper. It was simple, reflecting in his writings that something was wrong, that the puzzle had to continue, something like that. So I was not mad myself because the data are very clear enough, but if this is what happens here, you have to find out where in the brain the neurons have the capacity to process more than one modality in this test, and in what form. And this is the third part of my talk, and I will be ready for any open question about this. For over the years, we have discovered, shown from unimodal processing, that all these areas in the premotor network, ventral premotor cortex, medial premotor cortex, prefrontal cortex, dorsal premotor cortex. Here is where Risolati claims that we have a neural apparatus that allows us to understand others, mirror cells. And all these areas have been considered as premotor areas, just simply premotor because they are in front of the primary somatosensory cortex, which is here. And they were classified, these areas are premotor cortices, just simply because when the study of these areas began many years ago, they had a very simple motor test. And they showed that they had something to do with movements, not in the same way as the primary somatosensory cortex did. So for many years, these areas were neglected, that they could do some other cognitive functions. Just simple, the whole community started to produce very sophisticated motor tests in order to prove that these areas were motor, premotor. For example, motor planning. Even there was a Nobel Prize that probably you don't know his name. His name was John Eccles, who died about 15 years ago. That when he left science, he started to do, to think about consciousness and some other things. He was very Catholic. And he And by the end of the 70s, 1970s, there was a very interesting investigator in California called Benjamin Libet, who started to do some recordings, EEG recordings in the brain in paradigms where simple asked to the subject to do movements when they wanted, and related this EEG activity with the timing of the push press, and discovered that signals from the cerebral cortex started to develop well before the muscle contraction, and of course, well before the button press. And when he asked to the subject about in what moment they felt the necessity to begin the voluntary action, he discovered that signal was even before the muscle contraction. And so that led him to think that every voluntary action was involuntarily generated in our brain, and that every conscious action was unconsciously generated, which is becoming a paradox, because we feel that we made decision by our own. Maybe yes, maybe none. I decided to come to Barcelona. I decided to speak. I'm deciding to speak. But the claim, Libet claim, is that everything is developed in well before we make conscious, and our conscious is an illusion that we made decision by ourselves. In other words, that we are puppets of our neurons, and the two processes are epiphenomena, if you like. That's Libet. Believe me or not, I have my own thinking about this. It might be close to Libet, it might be not. In the 80s, Wolfram Schultz did experiments trying to find out the neural signals associated to voluntary actions, and trace a very large circuit in the brain in which signals developed well before the monkey made the first contraction to initiate a voluntary action. Even well before the EEG, because we were using single electrical signals. And we counted, Wolfram Schultz and I, the many loops, how many times a signal had to travel from the cerebral cortex across basal ganglia and the thalamus, and it happens that it's about 25 times before there is the first neural signal associated to a voluntary action. And so very likely. It takes time, and in fact, there is a beautiful book from Benjamin Libet, published by MIT Press in 2005. The name is Mind Time. It was the last cognitive product by Benjamin Libet. He died 2 years later, where he develops all the thesis that is a matter of time. If it's a matter of time, probably that series of papers that Wolfram Schultz and I published by the end of the 80s of the past century, past millennium, maybe is something supporting that before any voluntary actions, our brain has to develop something and comes to a threshold, and then we make conscious that decision to make a movement. And sometimes you have the capacity, very short period of time, to stop it, let it go or stop it. Makes a decision, because any decision in life might have certain consequences. Okay, so continue with this. So Eccles, just to finish this story, Eccles developed a very dualistic position. It's very strange, in a strange way, because Descartes was developed a strong dualism. Also because the mind and the body. And of course, Descartes had no knowledge about neuroscience, but Eccles had some neuroscience knowledge about this. And he developed a very strange story in which these areas where Libet discovered those EEG potentials preceding voluntary actions, that this area was used, but unknown by the soul, something ethereal that used the brain cells like puppets in order to drive our actions and do something, our decisions. And he said, this is a very strange book, if somebody is interested. The Self and Its Brain, published in the, I think in the 80s, with Karl Popper, one of the greatest philosophers of science, in which they had a dialogue about these interactions between the mind and the brain and all those kind of things. And I do remember quite well when I was with Vernon Mountcastle once while I was training the monkey, and I was sitting because the monkey didn't want to work. So I opened that book, started to read, and Vernon Mountcastle came to me and said, "What are you doing?" "I'm reading this." "Throw this into the trash. That's bullshit. That's not possible. You read it. Jack is crazy." And all this crazy, I said to myself, I was young at the time, and I felt a little bit intimidated. And suddenly came Jerry Edelman, too, another Nobel Prize, and said, "Oh, Jack is totally crazy." I had another idea. By that time, Edelman was writing a beautiful book, also very provocative book, called Neural Darwinism, in which he developed an idea in a Darwinian way, in which our brain cells are competing with all brain cells with the urgency, necessity of processing cells, and developed a very interesting story that ended in another interesting book by Vernon Mountcastle and Edelman called The Mindful Brain, in which Mountcastle proposed the model organization of the brain based on columnar organization, and from columns emerge distributed system, which in some way is what I'm doing, which are made for a distributed function. In other words, that there is no one single area in our brain which is devoted to something very specific. You need the coordination of brain cells for many circuits in order to produce a function. And this is the subject for Gustavo and for me and many others, and we are trying to understand how brain cell communicates in a given moment in order to produce something, to make how neurons synchronize or some other signals, how they communicate, large number of neurons to communicate in order to produce a signal. This is a very open question too, and I think it's totally open because we know tiny things. In spite of that, I continue. Okay. So these are unimodal. These are not unimodal. That will be the message. And I will take only this example because it's the most, it's a more rare area in which the data I'm going to show you can produce something like this. So the monkey, you simply let the monkey do the task, the bimodal test, and insert your microelectrodes and do the recordings of the cells, and just simple get the signal. So on the left, there is a neuron which is responding as a function of the stimulus frequency for one modality, and another neuron responding to another modality with the same stimulus parameters. And you will see, look at this, I'm increasing the stimulus frequency for either modality. You can see by eye, excuse me, that there is a modulation of the responses to one modality. This is tactile. The first is tactile here. The first is acoustic here. And you are increases the stimulus frequency here. And you will see that this increases a little bit during the stimulus presentation and keep something during the working memory component once the stimulus is gone for the tactile and for the acoustic. And you can plot, you can simply do this test, you see, and simply take the mean firing rate for each of these frequencies and just simply and get the slope, which is not only more than an index of the modulation of the firing rate as a function of the stimulus frequency. This is 0.86, which means that the neuron increases almost 1 spike by adding 1 hertz. In real life, it doesn't occur. It's simple and a statistical test. And take this. And for the acoustic, it's the same neuron. It's more or less the same. See? These are simple, the coefficients which come from here, and all these big dots indicate that the neuron is statistically responsive against the basal firing rate. So keeping a trace of the stimulus frequency as a function of the stimulus frequency of the tactile first stimulus or the acoustic modality. So by definition, these neurons are bimodal. Some, of course, as in life, humans are very different physically and in behavior. And neurons are too. And we do believe that we are puppets of neurons. Probably this is an indicator of this. Again, this is for the tactile modality. This is for the acoustic modality. You simply see the shape. This is by the end of the working memory component. Before the second stimulus presentation, this neuron is responding. It's not responding here. It's not responding here. Start to respond. If you were studying motor preparation or expectation for the second stimulus, you would say that this neuron is responsive, is reflecting in its activity expectation for the presentation of the second stimulus, or motor planning, which is more or less here. This may be true, you will see. Excuse me. May be true, but it happens that is modulated as a function of the stimulus quantity delivered here for either modality in the same way, working memory. It might be possible that this neuron carry a timing signal, expectation signal, or something like that. But on top, they carry information about this. Okay. This is very nice, and you do this regression analysis, you will see that just before the stimulus, look at the beautiful modulation for the two modality. Again, these are the coefficients indicating that even these working memory signals invades the second stimulus presentation, which is a requisite for the comparison process. So the two signals we had shown before, the two have to combine. And from the two signals, for the working memory signals and the stimulus signals, they are combined. And from there, it emerges a signal which is very consistent whether the second stimulus is higher or lower than the first one. Well, of course, these are positive driven, monotonic, positive monotonic. So they increase their firing rate as a function of the increasing stimulus frequency. But some other cells do exactly the opposite, like this. So they respond the most with the lower frequency and the least with the higher frequency, but always monotonically driven in such a way that you will have now a negative slope like this. Look at this. And it's similar for the two modalities. These are the regression coefficients, beautifully driven. So bimodal. There are two populations with the same input in that part is treated differently in the opposite way. We haven't gone far with this analysis with this, but we have shown before for the tactile modality that there are always two populations beginning in secondary somatosensory cortex in which these two populations treat the noise in such a way, and the fraction is very consistent with what in the way the monkey stores the signal in order to make comparison and decision making. We have to do And we haven't gone far away. What are the advantages for us for having bimodal or multimodal neurons in our brain to treat visual, acoustic, and tactile, anything in order to make decisions? So, but another interesting fact emerged immediately. These are all the neurons studied in taken with this analysis from this area, and the lines indicate the times at which the neurons have this monotonic signal during the stimulus and working memory component. The gray lines correspond to the bimodal, which are the, I think, 80% of the neurons. The blue ones are the unimodals, and the red, I think the pink or something like that, are the acoustic. But it's very interesting because first thing is that there are very few persistent neurons. They are simply the neurons encode and then decode, encode, encode. And then there are bimodal and suddenly they become unimodal or something like that. But by the end, most of the neurons become very demodal, which is exactly the necessity to make the comparison. Look at see 84 are bimodal, 7 are unimodal, and these are unimodal for the acoustic. So the network becomes very bimodal. So it doesn't care anymore about the modality. It cares about the cognitive function.
So Ranulfo, this is over all the cells you measured?
Yes.
So this is not a subset of cells that showed a response to the stimulus?
We did record 183 neurons, just simple stable neurons that could be studied enough with all the tests. That was a requisite for us. And then we simply took that neurons and found that practically all the neurons were responsive. And then we used very simple decoder. There is the multiple regression encoding.
So there are about 80 that are not amplified.
Exactly. And half of the neurons became responsive with this type of signals. Okay. Very few unimodal for the tactile, very few for this, mostly are bimodal. And now we are carrying one of the reviewers, because this paper is submitted to Neuron, we're in the second round, said, listen, but this is not the only way of decoding, of course. But this is something that we know quite well by sure for many years. And they suggested another type of decoding, in which, very likely, there are neurons that have some other shapes. Instead of having monotonic encoding, positive or negative, it might be possible that some neurons have preferences for one frequency or something. This is what happened. Doing a very simple ANOVA analysis, it happens, we recuperated 25 more neurons. So there are more neurons with that simple analysis, and some of those have an U shape or inverted U shape. Or, for example, they have preferences for one frequency. We don't know why. It might be artifactal, or it might be something that the network is doing, or probably the monkey at that moment was not very attentive. We have to go back again to the psychophysics or something like that. Well, let's, yes?
And those who are specific for a frequency are bimodal?
Well, I will answer that question first. So if you put the numbers as a function of time, it emerges a very large number of neurons. And the dynamics is quite interesting. I see how this is a trend to increase the bimodal by the time. Here is the comparison. A very few tactile and of the unimodal side. Just simple doing plotting the slopes from the values of the slopes, a function of beings for the tactile, the acoustic, or the bimodal, you will see that as a function of time, they lie almost in the diagonal. More beings are associated to the bimodal cells. In calculated information is exactly the same. Look at see how beautiful they go. Very consistent. Look at the number of beings for the bimodals or the unimodals is they grow up as a function of time, either for the slopes. Since they lie in the diagonals that they are sharing more or less, or represented the stimulus quantity, magnitude in the same way. And of course, these are the angle as a function of the angle. For example, thinking that 0 would be for the tactile and 90 would be for the other one, and those that lie in the center with the 45%, you will see that the beams start also to separate very nicely in this way. Such that here is at 45% by the end. This go for 37 from the beginning for the bimodal up to 45, whereas the other ones are very close to the extreme parts. And this happens in exactly the same calculated bits information. Beautiful. Now, the answer to Gustavo. For example, the question is whether, suppose that I have this signal as a function of the stimulus, acoustic stimulus frequency. Now, and then I use the tactile stimulus frequencies. Can I decode that signal from the acoustic responsive? Yes. And vice versa, in such a way. When you plot modality versus frequency, frequency is in this axis, modality is here. The colors are always the same, tactile, unimodal, acoustic, and bimodal. And you will see this interesting thing. Look at how it ends. It's almost impossible that you decode a signal from this as a function of the modality. So they become amodal, supramodal, they lose the capacity. So, of course, there is a question. Did we train these neurons to have this capacity? Probably yes. But at least we know where in the brain there is this plasticity, because this doesn't happen in the early sensory cortex. So, to conclude, so this is our tiny model, some model, very biological, the processing pathways for conversions of sensory signals into perception, memory, and decisions. We haven't gone into the comparison and decision process because it requires another type of treatment. It requires more analysis to use these working memory signals versus the sensory input and get the resulting component, which whether it's consistent or not with what the monkey is going to report, the decision report. So there is an open question here, what are the advantages of this bimodal cell versus the unimodal one? We don't know. My answer is that it will be very difficult to get an answer to this because we have very few unimodal cells. Takes me 1 minute to conclude. And the model is very simple. When you touch something, quantities, by the stimulus in this case, this area is ignited and represent the sensory inputs, stimulus parameters. When you deliver the auditory stimulus, it's ignited the auditory cortex. These are separate channels working, and probably at the same time. And an important question is, if I were to deliver both signals, are they competing at the same time? Which one is treated? What is attending? When you are in a crowd, you have acoustic, you have visual, and some other cues. What are they processed, attended at the same time? Yes or no? This is still an open question. We are running an experiment in which we can now deliver all these signals, but forcing the subject to attend only one. And my guess is that our brain has a very limited capacity to attend more than 2 modalities at the same time. It's an illusion that we can combine probably many things at the same time. It's more, everything might be delayed, but it takes time. When I say time, it might be milliseconds, but a time in which it's very difficult for us to consciously know. Then, once the stimulus is gone, this area is engaged, of course. It keeps stimulus parameters in working memory. Of course, all these areas too. Simply, I have no time to show. The data are there. And then when the second stimulus comes again, it has to be compared against this. And sure, the comparison process is not taking here, it's taking here. It's taking in all this network. So the question is, where does the signal, the instructive signal that drives the motor apparatus, the voluntary reaction of the animal, because the monkey has to do a voluntary reaction to press the push buttons and get reward, and how the reward affect this are open questions, of course. And this is the end. I hope that the message is very simple one. It's very neuroscience oriented. I think is get some knowledge of neuroscience. It's not bad for any person, I think. And this has the collaborators. Luis Lemos did the sensory part with the collaboration of Rogelio Luna, and of course, Jose Guevara did the second part with Natsuko, Roman Rossi, a Argentinian postdoc. Natsuko is doing an interesting experiment in which we are asking the monkey to tell us the truth. So we are giving an instructing signal. For example, we instruct the monkey that in the future there will be a tactile signals, but we deliver an acoustic. And the monkey has to tell you whether there was the acoustic, whether it was the tactile, or whether it was acoustic or not. And worse, we are delivering two sensory modalities at threshold level, which becomes a nightmare for the animal. But we are getting a very interesting bunch of data associated to the attentional component on how they treat different sensory inputs in order to make a decision, plus introducing a certain degree of uncertainty and removing the uncertainties from time to time in order to make decisions. I think is an important issue because our brains makes hypotheses about what's going on our side, and that's the reason why very often make wrong and correct decisions in one way or in another. So it's a pleasure to be here. Thank you.
Fantastic. So we have time for questions. All right, Ivan.
So thanks a lot for the presentation.
Very welcome.
Very simple question. What is added in the response of these primary sensory areas to the original stimulus in the periphery? So what has happened constructive, from the periphery to the thalamus to the cortex?
It's a classic question. When you go to the textbooks, they tell you how receptors work, how primary afferents, no matter what is the sensory organ and the modality, visual, olfactory, whatever, acoustic, tactile. So there is a transduction. The sensory organs become very specialized. It's a very interesting issue, how, for example, in the case of the somatosensory system, there are different cutaneous mechanoreceptors that translate, for example, they are very sensitive to motion, or some other to indentation. For example, the Meissner corpuscles are the ones that are basically active to the vibrotacta because it's moving the skin. It's exactly the same as when a mosquito walks in your arm. So this walking from one place is the signal that is interpreted that something is moving across your skin. Or for example, when you take a ball in your hand, those are basically the Merkel discs, which are associated to the force. So in this case, what happens in our stimulus is that when you touch the skin, there is the Merkel plus the Meissner are active at the beginning. And then there is a very fast adaptation of the Meissner corpuscles, but the Merkels are active, sending a signal. It's what is telling to the brain is permanently indenting your skin. And then when the motion signal comes, the vibration, then the Meissner again, and then that signal is treated by a channel into the brain. I want to believe that something is going on too also in the acoustic signal. So, beginnings, you need the receptors, and then there are very fast pathways into the upper part of the spinal cord, which is poorly understood. But I want to believe there is a very safe way. The synaptic process there is very safe in order to transmit the signal to the thalamus. We've been working in the thalamus. Even Gustavo is engaged in this business. What's going on? What the thalamus transmits to the cerebral cortex, because the cerebral cortex is blind. When you say this is the somatosensory cortex, there is nothing to tell you that this is the somatosensory cortex, simply because the inputs come from the thalamus. Because if you were to send the action of the thalamus to what the territory be for the visual cortex, that will be the somatosensory cortex, the territory. So there is a safe way of processing. What is not known is what's going on after that representation into the very vast network in the cerebral cortex for any modality.
All right, there's a question here by
Richard. Hello. So some theories of attention propose that what it does is it binds separate features of the stimulus. So what I'm thinking is, did you try to do this experiment when the monkey did not have to perform the task of comparison between the two stimuli, and see if the premotor cortex still represents these two features that are independent?
No, I haven't run that experiment. I've been the coding stimulus features from one sensory input, and I haven't gone into decoding more than two sensory modalities plus the features. There are many features for one single modality. My philosophy is very simple at the moment. I am 61, and we think we're going to live forever. And I have been calculated that probably I have 20 more years left in order to go into that problem. I don't know, it's a very interesting problem that attracts the most to me at the moment of my life. Probably will devote my time to that problem because I have known enough. Well, it's what I think. Everybody will tell me that it's not enough, man, as always. But I want to jump into that problem on the... So I clearly know that it's very difficult to study attention by itself, for example. That's the mechanism, because you have to think about what is a mechanism for attention. What I know myself and Gustavo and many people is that we can understand the effect of attention on something. And even the people thinking about consciousness, like Graciano, that published an interesting book recently, thinks that attention is the key way for understanding consciousness, because somehow being conscious or aware of something that happened in the external world requires of attending information. And what does Gustavo when he's alone and not depending on the external world, concentrated, is using his attentional resources to bring up information, and the key is information plus attention. So it might be, there is something interesting here.
So, Ranulfo, another question.
Yes.
When you look at these unimodal responses in the primary sensory areas, you could also, well, maybe that's also due to overtraining, because these monkeys are so hyper-specialized
Yes.
that what you maybe reveal is a learning process and not an intrinsic property of these primary sensory areas in the cortex.
Okay. Can I answer this?
Yes, please.
Try. If you take a naive monkey and then let him to sit in the chair and deliver for the first time the tactile stimulus, the primary somatosensory cell will respond very similarly to the trained animal. Second point. If you take this monkey, naive monkey, anesthetize it, and deliver the stimulus, you will get more or less the same responses. If you take the trained animal, and then in attentional passive way, you deliver the stimulus, the neuron will respond very similarly to the active mode. You anesthetize, it's still responding. There are tiny little differences in the phase log responses because you change a little bit the network in the active versus the attentive wave, and in being vigilant awake versus under anesthesia. I'm not claiming at this moment that these areas are very passive networks to represent information. Superimposed on this, there are these attentional signals. For example, the subject is attending something. And for example, the thalamus might be adding something, a gain factor, for the processing of the signal in the early sensory cortices. Probably those systems in the brain stem and in the diencephalon, cholinergic plus aminergic system, might be doing something important. But at the moment in these models, what my experience is that the networks which have a very high viability, by the way, very flexible, are outside the early sensory cortex. And I've been thinking about this. Of course, there is a learning, processing, and many other things over training. Of course, as I said from the beginning, you train the monkey. You come here to be train the network. So something might be happening, but I think that most of our learning doesn't happen there in the early. It happens after, downstream. And I have a very simple, just to end this. I think that if these networks, early sensory networks, have to be very rigid in order to preserve a certain rigidity, certain stability about the world or the representation of the external world into the brain. Because from every day, this where it changes, we have to run very fast a learning process in order to adapt. So these are systems that keep a reference, internal reference about the way we are going to interpret the external world. Otherwise, it could be a nightmare. And I think we need it, but this is simple speculation.
Mm-hmm. Because last week, when we were dealing with Evo-Devo, one example that was put forward is this idea of, or this phenomenon of echolocation in blind humans, right? Where also there's a correlation between activity in the primary visual cortex now and their ability to do echolocation, generating clicks. So there you would still see, also in adulthood, a plasticity of a primary sensory area that really makes it change its modality, or its commitment to the modality that the thalamus gives it.
Yeah.
So how do you explain that away?
I cannot explain because, as I said at the beginning of my talk, this can be a limited way of trying to address this problem of the where in the brain or whether early sensory cortices have this capacity. Again, in the Lemus paper, neuron paper, if you are interested on this, everything is there about this. We also add some signals, some responses in the early sensory cortex to another, not to the principal modality, but are modulation signals. But it's impossible to decode something about the sensory input. It might be possible that the non-principal modality might add something. And this is an experiment we are running right now, because of course the reviewers always say, well, you're using supramodal stimulus. You have to use subthreshold or threshold stimulus in order to see whether there is not something added. By eye, I haven't seen any, but again, I'm running the experiment right now. I hope the experiment is done, because once we go out, we don't know what's going on. Right. Yes, but this is, everything has to be open, because this is the role of science. There are new technological and conceptual development that need to be updated continually in order to see, to continue addressing this fundamental question that drives more of the principles to understand sensory organization from the neuroscience point of view, of course.
Right. Tony.
Thank you. It was a really
You're
interesting talk. So a couple of comments and question. So if I look at my hand and I have a tactile experience of my hand, then just the act of looking at my hand changes the experience of that. And Patrick Haggard, as I'm sure you know, has shown that you get a heightened response in S1 from the act of looking at a point where you're being stimulated. So you have a better 2-point discrimination, for instance, and that's correlated to activity in S1. And so there's evidence maybe of some modulation by vision of the gain, at least in somatosensory cortex. Michael Brecht has recently shown in rats that their whisker touch, if they use it in a social situation and they contact other rats, then the signals in S1 cortex resulting from that are modulated by the gender and other aspects of the social context. So for instance, if they contact a rat which is dead, they get a much lower response than if they're contacting a live rat, even though the stimulation might be the same. So these two pieces of evidence show that there's significant modulation of S1 by potentially stimulating other modalities, but also the broader context. In your particular paradigm, you have a very pure situation. So ecologically, we might say that this situation doesn't often occur where you have the signal just in one modality. And you could also imagine that if you present the tactile and the vibration and the auditory vibrations together, then that might give you some modulation of the response in the primary cortex. So at what point in your story, and this is my question, do you allow for some modulation of primary sensory cortical activity by other systems?
Oh, yes. Okay. That's the point, is from the beginning of my talk. I'm aware of this literature, and the problem in all of this is that the way is we pose the questions and we try to answer the question with conditions, experimental conditions. Sometimes I think, and I want to believe, that monkeys are different to humans and humans to the monkeys. And but because most of this literature comes from human studies and depending of techniques, massive volume recordings, which poses certain questions, versus single unit recordings, or at the most local field potentials. We've done analysis about the local field potentials, whether there is something, a rhythm, a signal there in the spectrum which is modulated by not the principal modality. We have delivered the two stimuli together, the acoustic and vibrotactile, and see whether the monkey improves performance. It doesn't improve because the monkeys are highly trained and are quite good to trade either modality, and threshold are basically similar. So I'm running some other experiment to see whether this. So I think it's, for many years, it's been known that the evoked potentials, the tactile evoked potentials can be modulated by visual. Jean Desmond discovered that since the 60s, 70s, something in humans, in a very well-controlled situation. And some P waves are modulated by attention and also other things. Something that I haven't seen myself with my preparation is, as I said before from the beginning of my talk, it probably it's very, there is something in the monkey brain, there is something in the way we train the monkeys, there is something in the task, there is something that doesn't go along with this provocative literature, is what I say myself. Secondly, coming to the social signals by Michael Brecht. You know, the whisker system is another issue because it's driven by the motor system, basically. It's a very complex organ. And as far as I know, although my mentor, Merriman Castle, wanted to say that the tissue, the cortex of the rat, is similar to the cortex of the monkey, and the human is not at all. Secondly, in the monkey, in the subhuman primate and us, the separation between early sensory cortex and the motor cortex are quite distinctive, although there is a strong connectivity. One drives the other and can drive some other things too. So in the rat, it's quite difficult to make a quite distinction between the somatosensory organ and the motor organ. They go together, and it's almost in free motion. So which makes difficult. I'm sure that if I were to let the monkey use its hand, it would be a nightmare to me to carry on this type of studies, because it would be extremely difficult to me to separate the sensory input from the motor component, plus some other cognitive things that are going together. So in an extreme, I'm using still a very reductionist approach in a very large system, whereas most of my colleagues do exactly the opposite. Very open things, complex stimuli to study tiny things. I use tiny stimuli to study complex things. That's my philosophy at the moment. It's been very productive, but it's been extremely difficult, for example, in the visual system to deliver complex stimuli and go into the visual cortex. Even the primary visual cortex is a nightmare to decode inputs, because from time to time eyes move here and there, and so the reference frame is complex. So I'm still running a very old approach coming from the physics and the chemistry in the 19th century in order to understand simple things. But of course, this is not the complete story. It's not dogmatic things. They are simple posing questions, simple questions, training the monkeys, and be sure with the psychophysics also produced in the 19th century in order to combine both and try to get something. But of course, I'm open to anything.
Thank you very much for a very interesting talk.
Thank you.
So my question is a little bit more philosophical.
Yes.
And actually, I got to be a musician, and primarily I have sensings connect to sound, but also I have been working with neuroscientists, and somehow I have some ideas about how sounds affects people. My question is that correct? That, let's say in this way, is that correct to think that when we agree about our sense, the different modalities are completely separate from each other? Is there, because there are signals, even sound signals, that are completely connected to other information? And how can we really be sure that the signal that we are producing with a sound, it will only affect the channel that in the part of the cortex that is connected to sound? Because for my expectation, for my experience, even sound, it has a very strong implication in feeling of tactile feeling, and even for smell, because if you think about time, but when you describe time. So for a psychoacoustic philosophical point of view, we would separate. But my question is that possible? I'm not talking only about the brain, I'm talking about the way the signal is produced. So the signal conveys other information by itself. So how hard, and somehow it's difficult for us to distinguish even in the signal. That's a little bit philosophical, but I guess...
No, it's not philosophical. Everything is philosophical.
Okay, thank you.
Okay? So that's the point. You made the point clearer than me from the beginning. The question that I'm trying to address myself is simple: where and how. That's the most we can do as neurophysiologists. So my answer at the beginning, because it's a very interpretative, is that it doesn't happen in early sensory cortex, but happens downstream. So the question is, let's think, for example, the arrival of the sensory input, somatosensory input into the cerebral cortex, it takes less time than the arrival of the acoustic sensory signal in the early auditive cortex. So from there is a time difference, okay? Then, being aware of something takes time, because the networks, even if it can be exactly the same network, there have to be an instructive signal coming from those early representations into the network. But also, the network is aware, making interpretation about those representations is like an observer, a metaphorical figure, observing the representation there. And when the attention of that observer fades away, that is, you don't pay attention to that representation, something might happen there. So the question is, where in the brain we gather, put together the different inputs. And the question also, which is fundamental because it's the way we interpret our inner life, how one modality, like a musician, for example, can be affecting, not affecting, that's not the correct word, is modulating or tuning some other sensory inputs too, or the interpretation of that. Not even that interpreting, but also producing something internal. Everything is internal, of course, but it's caused by other modality and touching part of the network which are producing this subjective experience of other modality. Those are open questions, and I hope very much that people coming from different disciplines and even from the neuroscience, different approaches will give an answer to this. But this is the point, and you made it quite well.
Like, for example, in music instrument playing, would it be possible that you are actually associating like a tactile sensation on the instrument and the sound that it produces that gives you this multimodal
you saw a tiny, very simple way of how to sense two quantities coming from different input sensory modalities can be treated equal. In Mexico City, one of my former students is doing more complex stimuli, of course, which is sometimes it's not very good for a beginner to use very complex models in order to study tiny things. That's my criticism to him. But he's treating visual inputs, very complex visual inputs, to be treated equal with complex stimuli, acoustic or tactile stimuli, trimodal, very complex ways. So maybe Luis can give an answer to this, but I don't know. It's been very difficult to do the psychophysics, human psychophysics, and of course, it's been a nightmare to train the monkeys because almost achieved the psychophysics. In human, he thinks he has something, but at the same time he's been for more than a year trying to train the monkeys when he didn't know the human psychophysics. But hopefully that's the way things go.
So Ranulfo, another question I would have is about the stimulus set that you're using, because by having these intensity modulated stimuli, you get an entrainment of these primary sensory areas, right? This is really an intrinsic property of these systems.
Yes.
So in some sense, this is a very strong stimulus that you're providing to the
Yes.
system, and you're exploiting in some sense one of these intrinsic properties. And this now propagates to a premotor area where it gets integrated. And what you showed in your last analysis is that in some sense, the modality specific elements get lost, and what you have left is more like a frequency response, or a frequency related response that sort of evolves over the 3-second time window of this memory field. But now, imagine the stimulus is not frequency modulated. In some ways, you're really hitting the system with a hammer, right? But now imagine we take a stimulus that's much more transient and more ecological, so that you don't have this really systematic modulation of the signal. Would you still expect a similar kind of dynamics in these premotor areas?
Okay. Let me try to answer. As always, we never have the answer in our brain or in our hands. This is a strong stimulus, of course, 7 times the threshold. But you have to, we made by purpose, because we didn't want the monkey become worried about detection. We have to read out of the gene the detection problem. It's just very quite detectable. So we wanted that the monkey go into the stimulus and that. But now, the point is that in error trials, the signals go away from the stimulus is there in the representation of the stimulus in the auditive cortex, primary, or in the primary somatosensory cortex, but never reaches the signal, the frontal network, and part of the parietal network too, when the monkey makes mistakes, errors, trials. Worse, control trials. For example, you can guide with the same stimulus, you deliver the same stimuli, the same test, but now you gave the monkey at the beginning of the trial a visual cue to tell him, listen, you don't have to worry to the stimuli, you simply know what push button to get the reward. Those responses go away. Now, if you put the monkey in a passive condition in such a way that the monkey will never press the push button to get reward. You simply deliver the reward from time to time, here and there. Those responses never occur. But the auditive cortex and the somatosensory are very active, responding to the stimuli. So that's the reason, that's the way. There is a gate signal, a gate that makes the transition from the early sensory representation to those areas. And the way is, why does the frontal lobe has to be aware of something where there is no need? It could be a waste of energy, driving the network continuously with the sensory input, there's no need. So that's where attention enters in action, to open and close gates. And that's economy. So why to come walking from the airport to here when there is a taxi? So it's exactly the same in our brain. So there are many gates here and there, and we need to understand that logic that makes, and of course, we have many words. We have terms from the psychological, neuroscience, computational, anything. So we have, at the moment, we are in an early stage where we have to put all together to know what we are trying to understand from technological, from the neuroscience, psychological, et cetera, and social too, which social business is becoming extremely interesting too.
Sure, absolutely.
Yeah.
Okay, Ranulfo Romo, thank you very much.
Thank you. You're welcome. It's been a pleasure. Thank you.